Welcome back to the Deep Dive. Today, we are opening up a file that challenges, well, decades of global safety regulation, touches every single person on this planet, and has some truly profound economic consequences. We're asking a pretty provocative question. Is the conventional wisdom about radiation risk fundamentally wrong?
Is every dose of radiation from a flight to a medical scan, is it all inherently harmful?
Exactly. Or can tiny doses, and I mean tiny, actually trigger protective, even beneficial biological effects?
And that's the deep dive today. It's this paradigm clash that drives all of our global radiation policy.
Right.
The fight between the extremely conservative linear no threshold model, we call it LNT, and this revolutionary idea known as the radiation hormesis hypothesis.
And these two ideas, they're completely mutually exclusive, right?
Right.
Yet both sides claim to have strong scientific backing.
They do.
The sources we've pulled together for you today are pretty extensive. We're looking at high-level epidemiology, dense cellular biology studies, even some really surprising clinical trials.
And they all offer this compelling thread of evidence that low-dose ionizing radiation, or LDIR, it seems to trigger a really complex, dose-dependent, and sometimes actively protective response in living things.
So our mission today is to really unpack this conflict. We need to understand the bedrock of current policy, what we all live under.
And then examine the detailed molecular mechanisms that seem to contradict it, look at the messy, real-world population data.
And ultimately, calculate the massive societal and economic costs that are tied to this one single unresolved scientific debate.
Yeah, and for context, when we talk about doses today, you'll hear us use the term milligray or MEAG.
Okay.
Just to make that meaningful for you, the average person in the U.S. gets about 3 milligay per year, just from natural background sources.
So 3 milligay. Keep that number in mind.
Yeah, keep that in your head as we discuss some truly eye-popping exposures later on.
Okay, let's get into it. Before we even touch on the idea of radiation benefit, we have to establish the global standard. The law of the land, which is this LNT doctrine. How did this one idea become the sort of unquestioned foundation of international safety policy?
Well, the LNT doctrine, the linear no-threshold model, it's the standard for radiation risk everywhere. And the definition is, it's pretty simple, but its impact is just enormous. It assumes that the risk of getting cancer or other bad health effects just proceeds in a straight line, a linear fashion, and critically, that there's no threshold, no safe dose.
So let me see if I've got this straight. If a massive dose of, say, 1,000 milligauss causes X amount of cancer risk,
Okay.
the LNT model just dictates that a dose 1,000th of that, just one LP, has to cause X divided by 1,000 risk.
Exactly. It scales down indefinitely and never hits zero. The risk is always positive, always there.
Which means any dose, no matter how ridiculously small, carries some defined, quantifiable probability of hurting you.
That's the core of it, and it's an extrapolation. We have to be clear about that. The model is based mostly on data from very high-dose exposures.
Like the Japanese atomic bomb survivors?
Precisely. In that group, the cancer risk is clearly linear and proportional. LNT just assumes that line continues all the way down to zero dose.
Even though the data for those super low doses is, statistically impossible to even verify?
It's lost in the noise of everyday life. You just can't measure it definitively.
And this isn't just some theory in a textbook. This is the official regulatory foundation for the world's most powerful bodies.
Absolutely. We're talking about the International Commission on Radiological Protection, the ICRP.
Right.
And in the U.S., the National Research Council, the NRC, especially in their big 2006 BERR7 report, LNT is the default setting for global safety, full stop.
And if LNT is the foundation, this is where it gets really, really expensive. The whole regulatory structure built on it is the LRA principle.
LRA, as low as reasonably achievable.
Which sounds sensible on the surface.
It does, but since regulators assume any dose is harmful, the logical, the ethical thing to do is to keep all radiation exposure as low as you possibly can.
Even if that means spending vast sums of money to achieve tiny, tiny dose reductions.
That's the rub. And the scale of the cost imposed by LRR compliance is, well, the sources estimate this one policy drives hundreds of billions of dollars.
Billions, with a B.
Hundreds of billions annually, worldwide. Think about a nuclear power plant. Every bit of shielding, every safety protocol, how they handle waste. It's all designed not just to be safe, but to be as low as reasonably achievable.
Which affects everything down to the lead apron at the dentist's office.
Exactly. It affects medical imaging, training. Critics argue we are just spending these vast resources to mitigate risks that, might not even exist.
And diverting that money from proven public health dangers.
That's the argument, a huge economic consequence based on a scientific premise that is, at its core, an extrapolation.
Okay, so let's play devil's advocate for a second.
Yeah.
Why do the regulators stick to this so staunchly? What is the core scientific defense for LNT, like in that ER7 report?
Their main argument really comes down to genetics and cell biology, how fragile it all is. The BA7 report just flat out rejects hormesis. It argues that for a true dose threshold to exist, a point where there's zero risk you would need, and this is, quote, totally error-free DNA damage response and repair.
And I'm guessing their evidence suggests that DNA repair is inherently not error-free?
Far from it. Precisely. They highlight the danger of DNA double-strand breaks, DSBs.
Which are catastrophic for the cell.
Absolutely catastrophic lesions in the DNA, and radiation causes them. They cite evidence that if you lose key DNA repair genes, like this one called M11-ish, the damage from radiation is massively amplified.
So their logic is the cell needs these genes to fix the damage, but the repair process itself is often flawed, error-prone, which could lead to mutations.
You've got it. So they conclude that risk has to exist even at the lowest possible dose. They're prioritizing prudence.
That's the crux of it then. DNA damage happens, repair is flawed, so assuming zero risk at any dose would be scientifically irresponsible to them.
And ethically negligent. They stick to the linear model because it's the most conservative, most protective estimate they have, regardless of all this other biological evidence piling up. For them, it's a necessary oversimplification for public safety.
Okay, so LNT gives us the baseline, this conservative assumption of universal, unavoidable harm. Now let's pivot to the counter-argument, the Hormesis hypothesis. This idea suggests the cell isn't just a passive victim, it's an active, adaptive system.
That's exactly right. And hormesis isn't just about radiation, it's a general biological principle. A low dose of something, a chemical, an agent, can trigger a beneficial response.
The complete opposite of the toxic effect you see at a really high dose.
A total opposite.
I'm thinking of an analogy, like 1 cup of coffee makes you productive, but drinking a whole gallon might send you to the hospital. The effect isn't just smaller, it's a different kind of effect.
That's a perfect analogy. And for ionizing radiation, radiation hormesis is defined as a stimulation, often beneficial, from low doses. So when you plot this out, you don't get a straight line. You get this non-monotonic relationship. It creates a U-shaped or a J-shaped curve.
Where the risk actually dips below the baseline in that low-dose zone.
The beneficial zone, yeah. Before it inevitably climbs up into the toxic zone at high doses.
That just sounds so biologically plausible. I mean, our sources point out this isn't a new idea. It predates all our modern concerns about nuclear power or X-rays. It seems like it's just deep-seated evolutionary biology.
It is. I mean, think about it. Life evolved in a radiation environment. It's always been here. Hormetic effects were observed way back historically in everything from plants where low radiation could actually increase crop yield to insects and vertebrates.
Showing increased growth, fertility, metabolism.
All of it. These organisms clearly developed ways to adapt and even thrive in ambient radiation.
Okay, so let's get to the human evidence, the good stuff. What is the most surprising study you've seen that suggests a beneficial effect on our health?
For me, it has to be the human lifespan data from the Japanese atomic bomb survivors. It's fascinating and so often cited by hormesis proponents.
Oh, yeah.
Researchers looked at people who were exposed to relatively low doses under 155 centigrade.
Oh, the SOGI is 10 milligrams. So 155 SOGI is 1,550 milligray. That's a huge single exposure, but still classified as low in that context.
Correct. And what they found was that this low-dose group, they actually had a statistically significantly longer average lifespan than the control groups who got zero measurable dose.
Wait, say that again. They lived longer?
Longer. The risk of dying from non-cancer causes, so things like infectious disease, heart problems, it only started to go up once the dose got above that 155 Sugoi mark.
That just flies directly in the face of LNT's core fear that every dose is a net negative. How could the body possibly turn low-dose radiation into a systemic benefit, especially with the immune system?
Well, low-dose irradiation has been linked over and over again to activating the immune system. We see things like faster wound healing in animal models, or increased resistance to toxins, and even tumor cell injections.
And what's happening on a molecular level?
It increases the production and circulation of immune cells, especially lymphocytes.
You mentioned a really elegant mechanism earlier involving T repressor cells. Can you break that down for us?
Sure. A key theory is that low doses might selectively destroy or reduce the population of these very radiation-sensitive cells called T repressor cells.
And these are the cells that put the brakes on the immune system, right? To stop it from overreacting.
Exactly, they're the brakes. So by getting rid of some of these brakes, the other T cells, the ones that hunt and kill pathogens and cancer cells, they just become more efficient, more active.
It's a differential killing. The low dose takes out the inhibitor cells, which leads to a net boost in immune surveillance.
A stunning example of biological complexity just trumping simple linearity.
Okay, so let's drill down even further to the molecular level. What specific adaptive mechanisms challenge the LNT view that all DNA damage is just inherently risky and error-prone?
Well, the LNT model relies on this assumption of proportional damage, but LDIR, low-dose radiation, it activates these complex, cooperative, protective processes. It's a whole different ballgame.
So how do they measure that?
Researchers can literally measure DNA damage and repair by counting what are called double-strand break repair foci in a cell, and these direct measurements show a fundamentally non-linear response.
And what does that nonlinearity look like?
It's not just that the repair mechanisms work a little better at low doses, they function significantly better. For example, at a dose of 0.1 gray, which is 100 milligauss, they work much, much better than at a high damaging dose like 2 gray or 2000 milligauss.
So the low dose is like a signal. It's a prep signal, kicking the repair systems into high gear.
That's a great way to put it. It slows the overall rate of genomic instability because any subsequent minor damage gets fixed accurately and super fast.
So the cell isn't just passively taking damage and fixing it badly like LNT assumes. It actively revs up its repair systems when it detects a low dose. It's like a molecular warm-up, improving its ability to handle both current and future damage.
Precisely. And we also see specific antioxidant effects. Low-dose exposure increases proteins like AP endonuclease, which is vital for fixing single-strand breaks. LDIR induces these robust antioxidative responses.
So it's activating its own internal defense enzymes to detoxify the free radicals before they can do any lasting harm.
Which points to a system that isn't passively accumulating risk, but dynamically adapting to minor stress. And that directly contradicts the BIA's fifth premise of universally flawed repair.
The theoretical stuff, the molecular mechanisms are just fascinating. They really show the cell's capacity for this adaptive response. But the LNT proponents, the regulators, they always come back to the same thing. Show us the proof in large human populations.
Right.
And that's why these high background radiation areas, HBRAs, are so critical. This is where nature is running the uncontrolled multi-generational experiment for us.
And the data from these places, it often provides the strongest observational contradiction to LNT, even though, it's inherently messy data.
Let's start with Ramsar, Iran. I hear that's a pretty extreme example.
Oh, it's the most extreme. Ramsar has some of the highest measured natural background radiation on the planet. It's due to radium and radon coming from local hot springs.
So what kind of exposure are we talking about for the people living there?
Some inhabitants are chronically exposed to doses that reach about 260 milliphi per year.
Whoa, okay, let's contextualize that number. The average U.S. background is 3 millivi. And occupational radiation worker is often limited to maybe 20 or 50 millivi a year.
Yeah.
So Ramsar residents are getting over 5 times the typical occupational limit year after year after year. The LNT model must predict just a catastrophic increase in cancer there.
It absolutely should. And yet the data suggests the opposite. Studies on the people in Ramsar show no significant increase in cancer mortality or incidence compared to their neighbors in control areas with normal radiation.
No increase.
And get this, some preliminary studies even hint at a decrease in certain cancer rates. They show clear radioadaptation, like no increase in chromosome aberrations in their blood cells.
So life, after generations there, has adapted to and maybe even harnessed this high radiation environment.
That's what the data suggests. It's a fundamental contradiction. It implies a dose threshold exists that is way, way higher than 0.
Let's look at another one, this time from Bavaria, Germany. Lower levels, but still elevated. What did the reanalysis of that population data show?
So researchers did this critical reanalysis of cancer and early childhood deaths across 96 Bavarian districts, all with different background radiation levels.
And they used a pretty sophisticated statistical approach, right? Bayesian analysis.
They did. And that was necessary because the original attempt to fit the data with a strict LNT linear model, it produced completely unrealistic results.
How so?
Well, the linear slope was so steep that it implied this mathematically impossible outcome, that over a third of the entire population would die of cancer if they were exposed to a dose of 1 sievert in a single year.
And 1 sievert is high, but it's an acute exposure that people can and do survive. The model was just producing this wildly exaggerated risk.
Utterly. When you extrapolate the LNT model down to these chronic low doses, it just proved to be a terrible fit for the real-world data. So the Bayesian analysis tested different ideas.
The linear model versus other possibilities.
Right. The linear model, a linear quadratic model, and a model of no dose dependence at all.
And what did that Bayesian test conclude?
The conclusion was that the model of no dose dependence, meaning cancer rates were just constant, regardless of the radiation dose across the districts, that model was about 7 times more likely than the linear LNT model.
Wow, that's some robust statistical evidence for a threshold or even a hormetic effect.
It is. It suggested zero correlation between dose rates and cancer or childhood deaths in these large populations, and it mirrors findings from similar regions in the US, China, and India.
This is powerful stuff.
Mm-hmm.
But this is also the point where the regulators, the LNT proponents, they just shut the door. This is the ugly side of the debate, the official critique of these HMRA studies.
It is. They raise 2 big issues, which leads bodies like the BEER committees to call these studies essentially meaningless.
And the first one is the ecological fallacy. Can you break that down for us?
It's a critical methodological weakness. You're comparing population averages. The average radiation level in the town versus the average cancer rate in that town.
So you can't track individuals.
You have no way of knowing the true lifetime dose and medical history of each person. You can't say for sure that person A's cancer wasn't caused by something completely unrelated, even if they lived in a high radiation town.
Right, and without that individual data, you can't rule out confounding factors.
And that's their second major point. Things like smoking, socioeconomic status, diet, climate, they're just notoriously difficult to control for in these big, backward-looking studies.
And those can have huge effects.
Huge. The sources point out that just variations in smoking habits alone can cause fluctuations in cancer rates of up to 40%. That completely swamps the tiny effect you'd be looking for from low-level radiation.
This brings us back to the radon paradox. The LNT risk estimates for indoor radon, the gas in your basement, they rely heavily on data from miners.
Yes, underground miners. And critics argue this is a fundamental flaw.
Why?
Because those miners weren't just exposed to radon. They were breathing in high concentrations of carcinogenic dust, diesel exhaust, all sorts of noxious gases in really poorly ventilated mines.
Which is a totally different scenario than a homeowner in a modern house.
Radically different. But the regulators, using LNT, extrapolate from that high-dose, highly polluted miner data to impose strict indoor radon limits, like the EPA's 4 picocuries per liter rule.
So the critique is, you can't use a dirty high-dose environment to estimate risk in a clean low-dose one.
Exactly. And what's wild is that when researchers like Dr. Bernard Cohen did these huge long-term studies with actual residential dosimetry across U.S. counties, they found the opposite effect.
The opposite.
Higher residential radon levels consistently correlated with a decrease in lung cancer mortality.
So the EPA's advice to reduce radon in your home might, if hormesis is real, actually be counterproductive.
Or even carcinogenic in some populations. For hormesis proponents, this is a prime example of regulatory dogma just overriding clean, definitive epidemiological data.
Okay, so the epidemiological data is noisy. It's subject to that ecological fallacy critique. To get around that, researchers have focused on controlled lab studies. These let us zoom right in on the molecular differences between how low-dose radiation hits healthy tissue versus how it hits a tumor.
Which is absolutely crucial if you want to know if this has any real clinical application.
Okay, so let's zoom in. One very specific study compared normal human bronchial cells, HBE, with A505 lung cancer cells. What was the difference in just basic cell growth?
The results were pretty dramatic. In the normal, healthy HBE cells, exposing them to low-dose X-rays, specifically between 20 and 100 milligauss, it actually stimulated them to grow.
It stimulated them?
Yes, the peak of this hormetic effect, this increased growth and vitality, was right at 75 milligauss. This is the classic low-dose stimulation effect in healthy tissue.
And if LNT were true, those cells should have been inhibited or damaged. But what happened to the A549 cancer cells at those exact same low doses?
Nothing. For the malignant A549 cancer cells, their proliferation was not affected at all in that same low-dose range of 20 to 200 milligauss.
So there's a differential response.
A huge one. Once the dose got above 200 milligauss, then both cell types started to decline, which confirms high doses are toxic to everything. But the implication is massive. Low-dose radiation selectively stimulates the survival of normal tissue while leaving the cancer cells untouched.
That suggests the cancer cell has some kind of vulnerability in its adaptive system. But the story gets even better when we look at protection, right? Did this LDR pretreatment protect the normal cells from a later catastrophic high dose?
It did. It confirmed the whole adaptive response idea. Researchers simulated a pretreatment. They gave normal HBE cells a little dose of LDR, 75 milligauss, and then they blasted them with a high therapeutic dose of 5 Gy.
And 5 Gy is a serious dose. That's 5,000 millibai?
A very damaging dose. And the LDR pretreatment was incredibly protective. It significantly reduced the toxic byproducts, the reactive oxygen species. It inhibited mitochondrial decline, and it strongly reduced apoptosis, which is programmed cell death. The LDR acted like a molecular shield for the normal cells.
Okay, I have to dig in here. You mentioned ATM and AKT phosphorylation earlier. Can we pause on that? What are these proteins actually doing? Are they like a master switch the cell throws?
They absolutely are the master switches.
Why does the cancer cell fail to throw it?
So ATM and AKT are kinases. They're critical regulators of how a cell responds to stress. When the cell detects that low level of stress from LDIR, it activates them. And this triggers this huge pro-survival signaling cascade, specifically something called the AKTGSK3-NRF2 antioxidant pathway.
So it's like throwing the switch that turns on the internal antioxidant and DNA repair factory?
That's a perfect way to think of it. And this is the critical finding. This whole complex protective mechanism was robustly activated in the normal HBE cells, but it was not activated in the A549 cancer cells.
That's the key distinction. The healthy tissue turns on its defenses, but the malignant tissue either lacks the right signals or just can't use that low-dose stress signal to survive.
Right. It suggests LDIR triggers these sophisticated defenses in normal tissue that cancer cells either don't have or need a much higher dose to activate.
Cancer cells are already so stressed out metabolically.
Exactly, and they often have mutated or busted DNA repair pathways. So the low dose is enough to make a normal cell stronger, but it's too weak to infect the cancer cell, which is already running on this broken, different engine, and that differential response is what gives this whole idea huge clinical potential.
We can connect this lab finding right back to the real world, to those high background radiation areas. The material had a study on melanoma in mice that used the extreme environment of Ramsar, Iran.
Yeah, a brilliant in vivo animal study. They housed these mice in environments ranging from normal background radiation up to 100 times normal background in Ramsar.
Wow.
Then they injected all of them with a very aggressive form of skin cancer, B16F10 melanoma cells.
So LNT would predict way faster tumor growth and lower survival in that 100X group.
The findings just shattered that prediction. The highest radiation group, the 100X group, showed the lowest rate of tumor volume increase over 24 days.
The lowest rate?
Significantly. The growth slope for the high-rad group was 0.420, compared to a really steep 0.807 for the control group. They even saw tumor size reduction and disintegration in the high radiation groups.
That's a staggering protective effect. What was the final survival outcome for that highest radiation group?
The survival rates were massively higher. They had 100% survival in the 100X background group at the end of the study period.
On 100%.
Compared to only about 55% in the control group. This is some of the most compelling live animal evidence that chronic high natural radiation can strongly impede cancer growth. It suggests a powerful systemic adaptive response is at play.
We've established that low doses protect normal cells, stimulate the immune system, and maybe leave malignant cells vulnerable. So here's where it gets really exciting. Can we harness this on purpose?
Yeah.
Let's talk about the modern resurgence of something called low-dose total body irradiation, or LTBI, in cancer treatment.
Right. So LTBI involves giving extremely low doses, often around 0.05 gray, so 50 milligauss to the whole body.
Yeah.
And it's not used to kill the tumor directly. It's used as a treatment primer. Modern research suggests it works mostly by stimulating the immune system and changing the tumor microenvironment. It makes other therapies work better.
How does it help the immune system in a cancer patient?
It seems to ramp up the activity of key immune cells, like natural killer cells, NK cells, and T lymphocytes. It also induces the release of certain signals that effectively change the area around the tumor from being immune suppressive to immune stimulatory.
So it makes the tumor a much easier target for the body's own defenses.
An easier target, exactly.
You also mentioned sensitization. This sounds like it goes beyond just an immune boost. It physically prepares the tumor for high-dose therapy.
That's the sensitization hypothesis. The theory is that LTBI stimulates the cells lining the blood vessels, but specifically within the tumor tissue.
And what does that do?
It causes them to secrete nitric oxide, or NO, and nitric oxide is a potent signal that causes vasodilation. It widens the blood vessels.
And why is having wider blood vessels inside a tumor so important for radiotherapy?
Because it increases the oxygen content of the tumor. And oxygenated tissue is much more sensitive to radiation than low oxygen or hypoxic tissue.
So LTBI acts as both an immune primer and an oxygen sensitizer.
Right.
It maximizes the punch of the later high-dose radiation that's meant to kill the tumor.
You've got it. Two-pronged attack.
Do we have any clinical reports that show this combination approach actually working in people?
Yes, there are some preliminary case reports that are very encouraging. One involved a 49-year-old woman with an aggressive ovarian tumor that had spread widely.
Okay.
She was treated with a combination of LTBI followed by local high-dose radiation,
And the result?
dramatic tumor size reduction, and even more, the tumor marker for ovarian cancer, CA-125, it just plummeted. Before treatment, it was way up at 140. After the combo therapy, it dropped to 35, which is inside the normal range.
Wow.
And other studies show that adding LTBI to local radiotherapy can increase the annual survival rate for patients with late-stage cervical cancer by 14%. These are really promising steps.
This adaptive response mechanism seems to be the key. The LDR is acting as a prophylactic, a shield, against the damage from the necessary high doses. Tell us about the rat study that confirmed this using damage markers.
This was a study designed specifically to test that countermeasure idea. They took rats, pre-irradiated them with a low dose of 0.25 G, and then 14 days later, they hit them with a high damaging dose of 5 G.
What did that low-dose pretreatment protect them against?
It protected them by actively counteracting the inflammation caused by the high dose. It blunted the rise in inflammatory markers like IL-2 and TGF-beta, and crucially, it significantly attenuated the rise in a DNA damage marker called 8-OHDG.
Which is a key signature of that oxidative stress.
A key signature. So the low dose acted as a systemic defense, mitigating both the inflammatory and the genetic damage from the high therapeutic dose.
It confirms that LDR induces these biological defense mechanisms that act as a countermeasure. This idea of preconditioning the body is it's the complete opposite of the LNT model.
Totally contrary. LNT would predict that adding that 0.25 G just adds more risk to the 5G. It doesn't mitigate its harm.
This sounds like a technology we never should have abandoned. The sources talk about a lost medical history. What happened to the common practice of using low-dose radiation therapy?
It's a fascinating story. The success of low-dose radiation for infections and inflammatory diseases, it just abruptly ended around 1948.
What kind of things were they treating?
Oh, things like gas gangrene, certain types of pneumonia, even inflammatory arthritis were commonly treated with low-dose X-rays, but it was supplanted almost overnight by the arrival of antibiotics.
The miracle drugs.
The miracle drugs, that and a huge shift in public perception after World War II and the dawn of the atomic age.
So the LNT model was just starting to take hold, along with the political climate.
Precisely. One source points out that for a lethal condition like gas gangrene, X-ray therapy was found to be the only efficient means of prevention and treatment in some cases, saving limbs and lives.
And yet this proven, effective therapy just vanished from medical practice.
Vanished. Driven by the combination of new drugs and this growing regulatory and public fear of all ionizing radiation, a fear that was strongly bolstered by the emerging LNT philosophy.
Which brings us right to the policy problem. If the biological evidence for hormesis is so strong, in labs and in the real world, why does LNT, the model of inevitable harm, remain this immovable, dominant global policy?
I think we have to look at the motivation. LNT is maintained primarily through what's called the precautionary principle.
And this principle, it sort of inverts the burden of proof, doesn't it?
It completely does. Regulators see LNT as the most prudent, the most ethical choice. They require overwhelming and conclusive evidence of a sustained health benefit or a clear, universally verified dose threshold before they will even consider abandoning the conservative LNT model.
So the burden of proof to change the policy is just exponentially higher than the burden of proof needed to question the science.
Infinitely higher.
And this rigid political stance, based on prudence, it has huge tangible consequences beyond just shielding nuclear reactors.
Critics argue the LNT concept actively promotes an irrational fear of radiation. And this fear has a massive economic drag. It hinders industrial development, especially nuclear energy, and it even restricts clinical care. Doctors are hesitant to use potentially helpful low-dose therapies.
And patients are anxious about necessary diagnostic scans.
Exactly.
Let's talk about the most tragic examples of this extreme implementation of the precautionary principle, specifically the evacuation policies after Fukushima.
This is maybe the most shocking cost of LNT adherence. After both Chernobyl and the 2011 Fukushima disaster, the policy mandated these massive, costly, and incredibly disruptive evacuations.
All to avoid radiation exposures that were comparable to normal natural background levels.
The policy was focused entirely on avoiding a minimal potential radiation risk.
And what was the measurable non-radiation cost of that avoidance policy in Fukushima?
The result was just devastating. The earthquake and tsunami itself caused about 20,000 deaths.
Nice.
However, the policy-driven evacuation that followed led to its own profound loss of life. According to the Japanese Reconstruction Agency, 1,632 deaths were confirmed from the stress, the disruption, and the trauma of the evacuation displacement itself.
These were not radiation deaths.
Not a single one. These were people who survived the disaster, only to die later from the stress of being moved, the loss of medical care, the total societal collapse in those evacuation zones, all in the name of avoiding minimal radiation.
The cure, in that context, was demonstrably and lethally worse than the disease they were trying to prevent.
It's a profound regulatory failure driven by a rigid, unthinking adherence to the LNT model. It's a critical policy inversion. The policy designed to protect human life ended up causing severe harm because it just failed to incorporate the complexity of human biology, sociology, and economics into its risk calculation.
It focused on a statistically uncertain risk while maximizing a certain immediate risk of disruption and death.
Sure.
Let's bring it back to the radon paradox, which is maybe the most personal policy problem for our listeners. The EPA tells people to reduce radon levels below 4 PCL based on LNT. If the Hormesis model is correct, what are the implications of following that advice?
Well, as we said, that original risk model came from heavily exposed miners, not people in their homes. So the LNT assumption leads the EPA to spend money and motivate homeowners to reduce radon, believing they're cutting risk.
But the actual residential data, Cohen studies, they showed the opposite correlation.
Yes. Those studies consistently found that higher residential radon correlates with a decrease in lung cancer deaths. So if we accept hormesis as possible, that low-dose exposure stimulates defenses.
Then actively reducing radon below those ambient levels might be counterproductive.
You might be stripping your environment of the very low-level stressor your body needs to activate those protective antioxidant pathways we talked about. The policy, driven by LNT dogma, could ironically be encouraging a state of partial radiation deficiency.
Which would make the policy decision itself carcinogenic, or at least harm-inducing, if the science of hormesis is right. So, if LNT is flawed, what's the policy recommendation from the hormesis proponents? What should replace LA-Aguirre?
The goal should shift completely. From a philosophy of just passive protection to one of active health promotion.
Okay.
Instead of just protecting the public against risk above background, the goal should be to actively promote health by setting conditions that reduce disease rates below background levels.
Meaning the regulatory limits should be set somewhere inside that beneficial zone of the hormetic curve.
Exactly. Proponents argue that exposure limits should be set within that beneficial, stimulating range of the J-shaped curve. By mandating a limit that's too low, way below that beneficial point, regulators are enforcing a lost opportunity for population health. They're denying the public the benefit of adaptive response, and they argue that is a fundamental policy failure.
We have covered a truly profound conflict today. On one side, you have global regulatory bodies staunchly committed to the linear no-threshold model, demanding we keep exposure as low as reasonably achievable.
And that's all based on the precautionary principle and the belief that because DNA repair is error-prone, any threshold is just impossible.
But against that consensus, we have this massive body of scientific counter-evidence.
Yeah, molecular biology shows normal cells have enhanced protection at low doses. They activate sophisticated error-free DNA repair. They trigger robust antioxidant defenses. They boost immune function. The nonlinear kinetics of DNA repair just fundamentally contradicts LNT's proportional damage assumption.
And then there's the real-world evidence, from epidemiology in places like Ramsar showing adaptation, to controlled animal models showing 100% melanoma survival in high radiation groups.
It all suggests that higher exposure might actually confer resistance to cancer rather than increased risk.
So the persistence of LNT appears to be more of an ethical and political commitment that imposes these massive societal costs, fuels irrational fear.
And tragically, as we saw with Fukushima, leads to unnecessary deaths due to overly conservative policies.
The sources really compel us to raise the ultimate provocative question then. Is ionizing radiation at low doses an environmental necessity for life?
The research suggests it meets all the criteria of an essential agent. There are studies conducted in radiation-deprived environments, like deep underground labs, that show when you reduce radiation way below ambient levels, animals can become weak and perform poorly.
Which suggests that we, humans, may in fact be living in a state of partial radiation deficiency. Our adaptive mechanisms are understimulated.
That's the key takeaway. If low-dose radiation stimulates the immune system and DNA repair, then the real challenge for future research isn't about fear anymore. It's about figuring out how to intentionally harness this adaptive response for cancer prevention and therapy.
Moving past decades of regulatory fear and into a more nuanced, maybe even beneficial future, if a low dose can act as a systemic countermeasure against the damage of a high therapeutic dose, then future clinical research has to move quickly to explore this. To run the human pilot studies, needed to find the optimum dose and timing to use this adaptive response for real clinical benefit.
Imagine intentionally using a tiny amount of radiation to make a massive therapeutic dose more effective and less harmful. That is the next great frontier in radiobiology, a fascinating convergence of molecular biology and urgent public policy.
A truly challenging deep dive. Thank you for guiding us through the science and the policy on this one.
My pleasure.
And thank you for joining us on the deep dive. We'll see you next time.